All right, so thank you all for coming. So today I am going to tell you a little bit about some new techniques we've been working on in the lab to image both cortex and hippocampus using microperiscopes. So these are chronically implanted glass devices that give us kind of novel windows of access to neural circuitry. So a little bit of just, very briefly, the kind of history of using microprisms and microperiscopes for imaging neural activity. So the idea is very simple. It's basically like using a periscope that you might see on a submarine, or this is actually a periscope that was used in the trenches in World War I for kind of seeing over the edge of the trench. And we use kind of a similar principle, except we kind of flip it upside down, use it to see down and into the brain. And so this was first done in Michael Levine's lab. And so here you can see one of these little microprisms. So this is a right angle prism, roughly the size of Abraham Lincoln's nose on a penny. This is a 1 millimeter prism. And they found that they could actually implant this into the tissue in the cortex. And what this prism does is it essentially reflects the imaging surface. So the hypotenuse is actually coated with a reflective aluminum coating, which allows you to flip your imaging plane. So the imaging plane will be perpendicular to the plane of the cortex. And when they did this, this was in a YFP-H mouse. So this transgenic line expresses YFP in a subset of layer 5 cells. And you can see this is their imaging plane here. They're getting these layer 5 cell bodies down at the bottom, and they're seeing their apical dendrites extending up to the peel surface. And they get relatively high resolution imaging. They can see, individual dendritic spines. So this was, just using kind of fluorescent markers in order to visualize the cells. But it was this first demonstration that you can do this in vivo and actually see these cells and track it over time. They made sure that it wasn't damaging the cortex too much. This gives a couple of advantages in terms of cellular imaging. So it allows you, as I just said, to image kind of transverse to the plane of the objective. Now this is really nice because a lot of the circuits we care about, such as the cortical columns, as I'm going to talk about later, the trisynaptic circuit in the hippocampus, these just happen to be kind of perpendicular at different angles to the plane of our objectives coming in. And the second advantage is that it allows you access to otherwise inaccessible regions. Just the same way that a periscope in a trench allows you to see out and over the edge of the trench, this can allow us to see down into deep regions that otherwise we would not be able to see from the surface. However, there are some major drawbacks to consider. So these periscopes or prisms aren't suitable for every type of experiment. They do cause some tissue damage because of the implant is going directly into the tissue. You're severing connections, you're potentially causing some inflammation, that sort of thing. Because you're imaging through quite a bit of glass and you're reflecting the image, there can be some modest reductions in the imaging quality. And I'll talk a little bit about the considerations, what we can do to kind of minimize the impairment of the imaging quality. Okay, so a few years later, people started, they saw that paper, and I was one of these people, and said, hey, we could use this actually image GCamp and look at functional activity in neocortex or other structures. And the first published work on this, to my knowledge, was from Mark Anderman, working also with Michael Levine and Clay Reed and others. And he imaged cortical columns in which they injected the neurons with an AAV to express GCamp3 or 5, I believe it was, and again, kind of use the same principle in order to see functional activity in all the layers of cortex. And they confirmed, this was nice because, maybe the neurons, they're still there, but they're dead or they're not responding normally. But in this paper, they actually confirmed that they're able to measure orientation tuning in the cells, and orientation tuning appeared normal. And they found orientation tuning throughout the column. They found the same kind of salt and pepper organization that had been seen from the surface. And they also did some characterization of changes in cell density, astrocyte proliferation, and found, as other papers and we ourselves have found, that most of the damage seems to be limited to within about 100 to 150 microns next to the prism face. So it's not like you're causing damage that's extending out for millimeters. So it's important to keep in mind because you want to try to image outside of the area that might be damaged. But what it does mean is that we're kind of limiting at least the acute damage from the prism implant And this is even, several weeks to months after implantation. So it seems to stabilize relatively quickly, and then we can image kind of deeper regions. Now, of course, this doesn't mean that we don't need to think about the damage at all, because we are severing connections. We're killing neurons that are kind of neighbors of the neurons that we're imaging from. So all of this needs to be taken into consideration when we're planning the experiments with these devices. Okay, and then I wanted to show you a little bit of work that we've done using functional imaging, but actually using microperiscopes. So when I use microprism versus microperiscope, the microprisms are just the right angle prisms, and the periscopes actually have the shafts, so they extend down deeper. And the reason we use this design is because we were able to then minimize the footprint. So the smaller you can make this footprint, the less volume the prism has, the less damage you cause to the cortex. So this was kind of the smallest we could get away with while still being able to image everything from layer 2, 3 down to the bottom of 5. We kind of wanted to not image the more superficial areas because there aren't a lot of cells there, and then they're just imaging the dura, things like that. So we really kind of focus on layer 2, 3 to 5. So this is a video from that region, and you can see, so up here at the top of the video, you have your layer 2, 3 neurons that are showing calcium events, and oops, sorry, it's supposed to loop, but at the bottom, you have your large, shoot. Okay, sorry, my video got stuck, but you can use your imagination. So at the bottom, you have the large layer 5 cells with dendrites extending up through towards the PL surface. And for kind of reasons of AAV tropism, the layer 4 cells are kind of hard to see, but if the video is working, you would see little smaller cells blinking occasionally as well. And it turns out we can actually determine the layers a little bit by looking at the ROI density as a function of the translaminar axis depth. So we see kind of an increase in density at layer 2, 3, and again at layer 5A. And so we can actually use this for doing kind of laminar determination. Okay. And another approach or another advantage of using these microperiscopes is that you are now imaging kind of within the plane of the somatodendritic axis in cortex. So for example, this is from a mouse in which we express GCamp in layer 5 cells using this RBB4 Cree line. And what you can see is that you have the cell bodies down here at the bottom, and these are all the apical dendrites extending up towards the surface. And you can see these are probably backpropagating action potentials, and they kind of reach all the way up. Here we were actually showing them drifting gratings, and we measured the pixel-wise orientation tuning. So each pixel, we calculated orientation tuning and colored it that way. So it's just kind of pseudo-coloring. And you can see each of the neurons kind of have their own orientation tuning, but you can also see orientation tuning actually in the individual dendrites. And some of the dendrites just share their somas orientation tuning because it's just coming from backpropagating action potentials. But if you look out here, actually, you can see that there's individual dendritic segments and spines that actually have different orientation tuning. So this could be used for, we're not really pursuing it ourselves, but this could be used for looking at dendritic integration during sensory coding or other processes. Now, the second advantage I mentioned using these microprisms and microperiscopes is the ability to access structures that are normally inaccessible.